So it's my pleasure to introduce our speaker today. Vladimir Kuleshov is a postdoc in Stefan Ehrman's lab in computer science at Stanford University. Prior to that, he received his PhD in computer science from Stanford University just last year. He was co-advised by Serafim Batoglou in computer science and Mike Snyder in genetics. And Vladimir's research lies at the most exciting intersection between machine learning and biology and medicine today. He's focusing on building machine learning tools to accelerate biological discovery and precision medicine, and fundamentally improving machine learning to make it possible. And his research has been featured in very prestigious biological journals such as Nature Biotechnology and Scientific American, Genome Web, and Science Daily. And his statistical phasing work has been licensed commercially by the Illumina Incorporated. At the same time, he has published many papers for top machine learning conferences addressing important problems in the field of machine learning. He's a recipient of the NSERC Postgraduate Fellowship and then the prestigious Stanford Graduate Fellowship that supports a very small number of exceptional PhD students at Stanford University. Today, he's going to talk about his work on accelerating scientific discovery in biology and medicine using artificial intelligence. Thank you, Brett.
Thank you. Okay, thank you very much for the wonderful introduction. So today, I'd like to talk to you about what I think is a really exciting research direction at the intersection of artificial intelligence, machine learning, and the biomedical sciences. So in the last 20 years, the pace of advances in biology and medicine has been truly astonishing. We are now able to predict diseases in a human fetus even before it is born. We have developed personalized treatments for cancer and for other diseases. And we're now on the verge of being able to easily and accurately edit the DNA of living organisms. These are truly remarkable achievements. But I want to argue that the next 20 years will see even more astonishing results. 10 years ago, the cost of sequencing a human genome was around $10,000 or more. Today, this costs less than $1,000. This dramatic decrease in the cost of biological experimentation will lead to massive new biological and biomedical data sets that will enable new discoveries. Currently in the US, for example, we have the Precision Medicine Initiative that is underway. And its goal is to sequence 1 million individual genomes and even full biomedical profiles about these individuals. At the same time, we have the Human Cell Atlas, which is trying to catalog the precise molecular mechanisms that are going on inside individual cells. These new data sets will lead to new scientific discoveries and to new medical technologies. And building tools and techniques in machine learning that make these discoveries and medical technologies possible is what I think one of today's most exciting research directions in machine learning and artificial intelligence. Now, this task will not be easy. This data that we're generating is going to be massive, complex, and unlike anything that we're used to. Just to give you an example, this slide here shows only a small fraction of the map of all the biochemical pathways that are going on inside a living cell. And in order to harness this complexity, scientists need tools that are powered by new machine learning techniques. And these tools will help them turn this complexity into useful scientific results. Building such tools and developing these techniques is the focus of my research. And this is what I would like to talk to you today. Now, the way that science has been done over the last several hundred years has been following the scientific method. And essentially, the scientific method says that we collect measurements about the world, which give us certain knowledge about the system that we're studying. And we use this knowledge to make predictions. which then lead to new experiments that we perform in order to verify these predictions, which forms a loop. I would like to structure my talk around the scientific method, especially focusing on the field of genomics. And I would like to present ways in which we can use AI and machine learning to make the scientific method, especially these two steps, more effective. And hopefully, by the end of this talk, I would like to have convinced you that there is ample opportunity to use the latest machine learning techniques in order to make science and discovery more effective. And in the first part of the talk, I would like to focus on the problem of biological experimentation. In this part of the talk, I would like to argue that scientific experiments can be made significantly more affordable and more accurate using techniques in machine learning in combination with the vast data sets that I have just described and that we're generating and that we will have much more of in the future. So this is going to be the focus of the first part of the talk. But before I go into detail, let me give you some brief background on the human genome. So some individuals have called, very metaphorically, the genome they have called it the book of life. So what it really is actually a sequence of about 3 billion nucleotides, which are the letters that we use, ATCG. And the genome contains instructions for how each of our cells should operate, which proteins we should be making, and when. Now, what you may not realize is that each one of our cells actually contains 2 copies of the genome. One is inherited from our mother, and another one is inherited from our father. And these 2 copies contain the same information. They contain the same set of genes. And this is nature's way of introducing redundancy into the system. If one copy of the genome is somehow broken, then we still have the other copy to provide us with some information. The genome also carries genetic markers, which are essentially mutations that confer us certain traits or that can predispose us towards certain diseases. So for example, this particular letter at this particular position could lead to lactose intolerance. And maybe another marker somewhere would make us have a different eye color. So these markers play a very important biological role in order to determine who we are. But what is really interesting is that not just the markers themselves, but their configuration on the 2 genomic copies also plays a really important biological role. And this configuration is something that we call in genomics the phase of the genome. And in order to explain what phase is, I'm going to use an example. So let's say we have here 2 copies of the genome, and one set of variants, which I have denoted in black, is on the blue copy. Now, in a different individual, we might find exactly the same variants, but the same markers, but these markers would be found on different copies of the genome. So now some of the black markers are on the red copy, and some of the white markers are on the blue copy. And a different individual might have the following configuration, and a third individual might have yet another configuration. These are the same markers, but they are located differently on the blue and the red, on the paternal and the maternal copies. So phase plays very often an important biological role. For example, phase influences our immune response. And so if we are trying to do a heart transplant to these individuals, maybe the first 2 will reject the organ, but the last one will accept the organ. And so phase is something that is, it's a fundamental characteristic of the genome that also plays an important biological role and is very important in many applications. However, most sequencing approaches are not able to resolve phase. Most sequencers will produce a single genomic string that will be a mixture of our 2 copies. And the problem of genome phasing is to determine whether the true configuration of the genome has this form or this form or this form. And again, here we have the same markers, but their configuration on the two copies is different. The best thing that we can get with existing technology is only a partial view into genomic phase. There exist techniques that can assemble the two genomic copies into short fragmented blocks. And these blocks give us some partial phase information. So for example, we know here that on the left, the C and the A are on the same block, hence they must be on the same genomic copy. And the same thing can be said on the right. But what we don't know is what is the global phase of these blocks? Is the global configuration like this, or is it like this? Right? We have these two options, and this information is not able to identify which one of these holds for this specific individual. So in this part of the talk, I would like to argue that the problem, this specific problem in genome sequencing, the problem of genome phasing, can be solved significantly more effectively and more affordably and more accurately using data-driven techniques. And I'm going to specifically introduce a phasing technology that, by relying on external data from past sequencing experiments, can make the problem of identifying phase significantly more effective and affordable. But before I go on, I would like to first pose the question of why is this even possible? What is the structure that is present in the genome that makes it possible to apply statistical techniques of any sort? It turns out that genomes have a very rich structure due to a biological process called recombination. And recombination is something that happens every time when a parent passes on their DNA to their child. So here I have two couples, and each of these, each parent in the couple will pass one copy of their genome to their child. Crucially, this copy will be a mixture of the two copies of the parent. And equally crucially, these two copies will mix only at a small number of points. And so the resulting genomes that will be transmitted will be a sequence of contiguous blocks that come from a different copy of origin in the genome. And I am denoting these using different colors. So this stretch came from this copy, and the light red copy came from the light red block came from the light red genome. Now this gets even more interesting if we repeat this process over multiple generations. Even at the next generation, the genome will start to look like a colorful mosaic of ancestral genomes, where the different colors represent stretches of DNA that have been inherited in as a single block from some distant ancestor way back in the past. And this structure makes it possible to apply statistical techniques to genetic data. For example, because all of the sequence on the dark green segment was inherited in one block, it is possible to say interesting things about this sequence based only on a few samples from it. Essentially, all these colored variants are very strongly correlated. And this makes it possible to do lots of interesting statistical analysis in this setting. Now, how can we use this information in order to perform phasing more effectively? Here I'm going to give you a toy example to illustrate the high-level idea. Let's say that we have access to a data set of phased genomes that was obtained previously using a more expensive sequencing technology. And these data sets exist and they are publicly available. And we have also a set of unphased blocks that we have obtained using the best available current sequencing technology. And our goal is to determine the global phase, the global configuration of these blocks relative to each other. Now, in this example, our subject individual carries a genomic sequence, which I'm denoting here using the color purple, that is shared with the first genome in our training set. And the reason that this might happen is because our individual and the first training genome share some distant ancestor way back in the past. And our subject has inherited this purple region from the same distant ancestor as the first member of our genome panel. And so what this means is that the most biologically plausible way to determine the phase of these blocks is to make the purple sequence go together. This is what is the most consistent with the structure that is implied by the phenomenon of recombination. And something similar can be done on the lower side of on the other set of blocks as well. And more generally, we can use our knowledge of genomics in order to define a probabilistic model, which will assign higher probabilities to a genomic phase that is more biologically plausible. And doing phasing will be equivalent to doing inference in this probabilistic model. Now, the model we will use will assume that there is some unseen latent phase genome from which our data was generated, from which those blocks were obtained. And this latent phase genome will be represented as a mixture of the genomes in the phase data set. And so in this setting, the most likely mixture is essentially the following. And determining this most likely mixture is an instance of an inference problem in a probabilistic latent variable model where this latent genome is the latent variable. Now, you might ask, why is this even possible? What makes it possible to, why is it possible that my genome can be represented as a mixture of a set of genomes of some random set of individuals? The reason is because humans are surprisingly related. And even if I take any moderately sized set of diverse genomes randomly sampled from individuals across the world, I will be able to represent my genome as this mixture in most cases. And so this makes this kind of analysis possible. Yes?
So are the phased data set is it known or not known?
The phase data set is known, it is available to us, and it's been obtained using some more expensive sequencing technology. And so this, for example, could be the 1,000 genomes data set, if you're familiar with what that is, or it could be another data set. What is also interesting is that we are formulating the problem of genome phasing using the language of probability. We're using a probabilistic model to determine genetic phase. And this introduces two challenges. On one hand, we need to define probabilistic models that accurately capture our understanding of the underlying biology, which in this case is recombination. And also, we need to define our model in a way that is computationally tractable. So there's an entire field of machine learning called probabilistic graphical models, which studies when it is possible to, that studies how to do inference in probabilistic models with a certain structure. And if you're familiar with that field, the structure that is used in our model is approximately the following. But the key idea is that this field describes a naive algorithm that can solve our optimization problem in a time that scales as k to the 4, where k is the number of genomes that we have in our data set. There exist also more advanced techniques which can solve this problem in a time that scales as k to the as k cubed. And in our work, we have built upon this earlier work by Garmani and Jordan, and in addition, we have used the specific structure of the probabilities that our model has, because these probabilities are derived, again, from some biological principle that have a particular structure. And so we were able to use this prior knowledge to do inference in a time that scales as k squared. And this is really important in applications where we might have a large set of genomes, such as more than 10,000. Now, how well does this technology work in practice? It turns out that it works surprisingly well. In 2013, when we started working on this project, there was a leading approach which was able to obtain a genome with a certain level of performance at a cost of around $8,000. At the same time, there was a technology that was developed in Stanford that could produce a phase genome for $1,000. But the performance, the length factor, was 10 times shorter. Using statistical approaches, we were able to match the performance of the state-of-the-art technology without increasing the cost. So we were able to get the best of both worlds. And so this is really important because now at $1,000, phasing is something that can start to become more routine. It is something that is becoming more affordable and can be used in standard, in a more routine environment. So we have published this first technology, this first paper in this first technique in Nature Biotechnology, and then later we had a set of follow-up works which again used the same principle of using statistical methods to enhance biological assays. In the follow-up paper in Nature Biotechnology, we showed how a variant of the phasing problem can be defined for bacteria that live in the human gut microbiome. And we were able to apply an extension of this technology that revealed new insights into the complexity of the gut microbiome. And later on, we also studied more general techniques based on neural networks, which are able to enhance various biological assays that are not necessarily related to phasing. But most interestingly, this technology was also later commercialized by the company Illumina. Well, it was first commercialized by a startup company spun out of Stanford, which licensed this technology. And later, this startup company was acquired by Illumina, which is the largest sequencing provider in the world right now. It's a multi-billion-dollar company. And because the cost of phasing was now at a level that was commercially viable, they deployed this technique as part of their genome phasing service. And so it is still used in their phasing product in order to offer this service commercially. And so I'd like to summarize this part of the talk by saying that we were able to take a sequencing problem, an important sequencing problem, which is determining genomic phase, and by using techniques that are statistical and data-driven, we were able to significantly reduce the cost of this technology to the point that it became commercially viable to deploy it in practice. And this result found interest from a company which deployed this service in the real world and made it possible to, and basically made this technology available in practice to most individuals. So this concludes the first part of my talk, which has focused on enhancing experimentation. Now, in the next part of the talk, I would like to focus on the problem of interpreting the genomic data that we have just obtained. So how can we use these genomes that we have sequenced in order to make useful biomedical predictions? Before I go into detail, I want to first ask again the question of what is even possible to predict from a genome? It turns out that we can predict many useful things. Our genome have information about our medical risk. They also influence our response to medical treatments. And recently, they have been shown to even contain enough information to make surprisingly good predictions of traits such as height or IQ or weight. In fact, the prediction from genomes has been named as one of the breakthrough technologies in 2018 by the MIT Tech Review. What is interesting about these problems, however, is that they're all intimately related to human health and well-being. And any techniques that tackle these problems have to be safe, they have to be reliable, and they have to be interpretable. And building these machine learning techniques that are safe and reliable is going to be the focus of this part of my talk. And as a running example, I'm going to take the problem of predicting medical risk. So we have a set of individuals here, and for each individual, we want to predict a probability of some medical outcome, which can be either positive or negative. And we're going to assume that each individual is going to be characterized by some features, which can be either a medical record or a genome. And we're going to apply a machine learning approach, and our machine learning system will be also parameterized by some free parameters or some weights W. And for each parameter W, the model will predict a probability. Now, the way that we build machine learning systems is by training them, is by providing them with a training set of labeled examples. So in our case, this would be a set of individuals with some known labels. and we find the weights of the system that give us the best performance on this data set. So here we can try the first set of weights, and it doesn't give us good predictions. The second set of weights gives us better prediction. Here only this individual is an error. And the last set of weights gives us the best predictions. And in order to define what is a good prediction, we define a loss, which is high when our predictions are far from the true labels. And the simplest loss we can think of is the absolute value difference between our predictions and the true labels. And we find the weights that minimize this loss. This is just any machine learning algorithm. And now we can take this algorithm and put it in the real world. And we can start seeing patients. And if these individuals are similar to ones that we've seen before, we will be making accurate predictions. This is the fundamental principle of machine learning. If our training data is similar to future data, we will be making accurate predictions. However, in the real world, the data that we observe can change. And so we might start seeing individuals which look unlike anything that we've seen in our data before. And so making accurate predictions on this data may not be possible. And this is a fundamental problem that is present in machine learning systems. And it is perhaps one of the main challenges for building safe and reliable machine learning systems. And so in this talk, I would like to, I'm going to describe systems that have guarantees even in the face of changes in the data distribution. Now, how can we achieve such guarantees? Well, one approach is to allow our system to learn continuously as it sees more data, even after it has been deployed in the real world. And so before, this was our workflow, and now we're going to assume that the system can eventually observe the true outcome and use it to update its state. And this can be done in our setting if we track the outcome of a patient over time. And if we unroll this loop, we get something like this. A system makes a prediction. It later observes an outcome, which is fed back to the system, which gets a new set of weights. We make a new forecast. We again observe an outcome. The system sees the outcome, makes a new prediction, and so on and so forth. If we plot the parameters of the system over time, and here I'm denoting different parameters with different shades of purple, we see that the system will gradually change its internal state as the data is changing. Now, it is one of the most remarkable and surprising results in machine learning that it's possible to obtain good guarantees in this setting, even under very, very general changes in this distribution. And to make things concrete, let's first define what it means to make good predictions. Let's assume that we have a second learner called the batch learner, which has seen all this data so far, and it has seen the true outcomes of these individuals, and is going to choose the single best set of weights that will minimize our target objective, our target loss over this data set that we have seen. So it's really surprising that there exists algorithms that, given enough data, can match the performance of the batch learner in the online regime. This is a really surprising result, and why it's surprising is because this is often possible for very large classes of models in very general settings, even when this data can change arbitrarily. So the data could be correlated, IID, it could be even chosen by an adversary as far as we know. And in fact, there's an entire field of machine learning called online learning, which studies this problem. And so these are some examples of textbooks from this field. There are many important papers in this field. And essentially, this field studies settings when it is possible to obtain guarantees of the form that I just showed. And it looks at what objectives we can use and what kinds of models we can use. It looks at convergence rates, etc. Now, I want to argue that this is not enough in many settings. And to explain why, I want to tell you, I want to give a short story. So you may have heard this story. It's actually, so it involves the actress Angelina Jolie. And in 2013, Angelina Jolie wrote a very controversial column in the New York Times where she explained that she has recently learned that she's carrying a rare genetic mutation called BRCA1. which significantly predisposes her to developing breast cancer. And in order to avoid developing cancer, she decided to undergo a preventive mastectomy. And in this controversial article that received a lot of criticism later on, she outlined the reason for why she did this. And if you read the article, she says things like, "My doctor told me that I have a risk of 87%, and typical women have a risk of 65%. And so after thinking about it, I decided that the best action for me would be to take this procedure." What this is showing is that in important real-world applications, it is not enough to just classify examples. What would have happened if the doctor gave her a probability of 70%? Would she still have done the procedure, or would she have chosen a different treatment? Would she have seen a different doctor? What the story is showing is that in real-world applications, it is important to give predictions that accurately model our beliefs and our uncertainties about a given problem. And these must be reliable and safe, and also must be formulated in a way that we can use them in order to inform, that we can communicate effectively patient risk, and we can use them to choose medical treatments in a way that is most appropriate for the patient. And so in this part of the talk, I want to look at ways in which we can obtain these algorithms in in which we can obtain such predictions in machine learning. And but before I go into detail, I just want to point out that typical machine learning methods don't provide such predictions. Here's a simple example. Here we have 4 individuals, and we have a set of predictions that achieves a respectable accuracy of 75%. However, these predictions are overconfident. We're saying that we're 100% sure. A much better set of predictions would have been 75%. However, it would be better because the actual success rate is 75%. However, this set of predictions happens to have a higher absolute value loss than the overconfident predictions. And so this just shows that standard machine learning approaches may not give us the kinds of uncertainty estimates that we need. And so I would like to describe an approach that will provide these uncertainty estimates in online learning, and in fact in machine learning more generally. And the approach I'm going to take is I'm going to define a framework that can take any online learning algorithm and transform its predictions in order to make them compatible with, in order to make them accurately estimate uncertainty. So the setup is going to be as follows. There's going to be a baseline predictor, which can be any online learning algorithm, or it can even be a human, and it will make a set of predictions that will go into a transformer module, which I'm going to define. And the transformer module will output predictions that are better in the sense that it will capture our uncertainty better. And then this will, again, be in this online regime. So what qualities do we expect of this transformer? Well, one thing we obviously want is the transformed predictions should be at least as good as the original predictions in terms of the loss that we're trying to minimize, given that we have seen enough data. And also, we want to quantify uncertainty. And to quantify uncertainty, we're going to use a simple notion called calibration. Essentially, what calibration is saying is, whenever I have predicted 60%, if I go back and I look at the actual success rate when I predicted 60%, it should be approximately 60%. So the predicted and the observed success rates should match for each set of predictions. And I'm also going, I forgot to mention this, but I'm going to make the technical assumption that the forecasts we're going to be making will be discretized. So it will be, say, 60, 70, 80%, 90%, et cetera. So these are 2 natural goals. How can we build systems that have these 2 properties? And so in order to explain the idea, let's first do a warm-up. Let's assume that we have some binary sequence y, which is not even related to any features x. And let's define the following simpler setting in which our goal is to make calibrated predictions of the sequence of this, just the simple binary sequence. So we're going to have a method which I'm going to call a subroutine because we're going to use it later, which at every step makes a prediction, then nature reveals an outcome, then we observe this outcome and make a new prediction, then we observe another outcome, and we go back and forth like this. This is the same setting, but now we don't have any features anymore. What is really surprising is that in this setting, we can also obtain calibrated predictions, even when these outcomes are chosen, can be chosen arbitrarily. They don't have to be tied to any distribution. So this is a surprising lemma that was first shown by Foster and Vora, and in our work, we gave a different argument for this lemma that enables us to prove some additional results and also to reuse the subroutine in the original problem that we're trying to solve. And I would like to give you a short argument for why this is even possible. I'd like to give you a brief intuition. So in order to show this lemma, we're going to combine two different ideas. One comes from probability, and the other one comes from optimization in game theory. And these are different fields, but by combining these ideas, we're going to be able to prove this lemma. And so the first tool we're going to use is the one that comes from game theory. And it's something that's called internal regret minimization. Essentially, internal regret minimization is a clever way of minimizing a loss function. So let's say that we have made a set of predictions, and now we go back we retrospectively look at our predictions, and we say that the learner has minimized internal regret if we never want to change our predictions of 30% to something else, such as 60%. Predicting 60% would make it worse. So this is a property of our predictions that we can obtain if we use certain algorithms that were developed in game theory. It's a clever way of minimizing the loss that gives us this property. And so this equation just shows the same idea. At all the times when we predicted P was actually the best decision that we could have made in retrospect. So this is a simple concept. And we're going to need one more simple tool, which is going to be which is something that we call proper loss. A proper loss is a loss function that has the following simple property. If we have a set of outcomes, so we have a set of binary outcomes, then the single probability that minimizes the average loss at these predictions is, what should it be? It should be just the empirical rate. So because here we have four positive and 4 negative outcomes, the single probability Q that minimizes the average loss at these time steps should just be the empirical success rate. It should be 50%. This is a simple property of a loss that we're going to use, but not all losses have it. So for example, the L1 loss doesn't have this property, the hinge loss doesn't have this property, but the squared loss has this property. And now using these two tools, we can show, again, this surprising fact that it is possible to make calibrated predictions, which is a very kind of probabilistic notion, under general distribution-free assumptions. And our construction will be to run an internal regret minimization algorithm on a proper loss and then we're going to target this binary sequence. So internal regret minimization tells us that P was the best decision we could have made at times when we predicted P. But because L is the proper loss, the best decision at times when we predicted P is the empirical rate at times when we predicted P. And so these two arguments are exactly the same. And so what this is showing is that this approach will produce probabilities that are calibrated. So P will equal the empirical rate, which was just our definition of calibration. So this is a surprising result. And we can use this result we can use this subroutine in order to achieve our goal that we had initially. We can use it to build systems that make predictions that are both accurate and calibrated when accuracy is measured using a proper loss. And the construction will be as follows. I'm going to instantiate. So first of all, this baseline, again, it's making a set of discretized predictions. This is a technical assumption, again, that we need in order to do this analysis. And for each possible prediction that it could be making, which could be either 80% or 60% or 40%, I'm going to instantiate this subroutine. And then when the baseline predicts a forecast of 60%, I'm going to fire up the subroutine that says 60%. and it's going to make some prediction. We will observe an outcome, and we will feed it back to the original, to the subroutine. Then maybe at the next stage, I'm going to make a prediction of 80, the baseline will make a prediction of 80%. We will fire up the subroutine for 80%. It will predict 90%. We observe the outcome, and we update the subroutine. So this construction uses our previous lemma, and it will have these two guarantees, calibration and minimizing the loss. So it will be calibrated because each of these subroutines is calibrated. And it will also be minimizing the loss for the following simple reason. So if we take each prediction, if we take each subroutine, then this subroutine has to be more accurate in terms of minimizing this loss than the baseline when we invoke this subroutine. But we invoke the subroutine when we predict, when the baseline says 80%. And so these recalibrated forecasts have to be more accurate than just a constant prediction of 80%. But the subroutine uses this really clever way of minimizing a loss, which is called internal regret. And so it turns out that this complex way of minimizing an objective function or to minimizing our loss is better than just predicting 80% constantly. And so this is why this construction gives us both accurate and calibrated predictions. So this is the theory. Now, how well does this method work in practice? What can we get from it in a real-world setting? So here I have an experiment where I am predicting the risk of diabetes on a set of 7,000 patients that come in one at a time, and the features are a set of 500,000 mutations. If we apply a baseline logistic regression algorithm, this algorithm will gradually minimize the loss. And here I'm using the L2 loss to measure the accuracy of predictions. And this loss will gradually go to zero, will gradually go down, and it will plateau at some value. And the reason is because we just cannot make perfect predictions. We don't have all the data we need in order to be perfectly accurate. If we use our approach, then initially we will be less accurate. But after seeing enough data, we will match the accuracy of the baseline predictor. Now, we can also look at the ability of our forecasts to estimate uncertainty. And we can look at how calibrated they are by defining a notion of calibration error. And this is basically how far are the observed rate different from the predicted rates. And the calibration of the logistic regression initially goes down and then stabilizes at a fixed value. But now if we apply our method, then as we observe more data, the predictions become perfectly calibrated. And again, this is guaranteed to happen because this is guaranteed to happen even under any distribution, because our algorithms have been designed to be robust to any changes in the distribution that we're seeing. And also, in a way, this is kind of it's a really good thing, because the only thing we have paid in order to obtain this good uncertainty estimation is an initial loss in accuracy at the beginning of our training procedure. Yes?
So I want to understand this method for internal regret minimization. So let's just, in the context, for example, when I flip a coin and I prediction 50-50 and I saw 3 heads in a row, does this mean I'm going to be more likely to predict tails next time?
If you've seen heads more?
If you've seen 3 heads in your prediction, so yeah, I'm just wondering.
No, well, I guess you would probably be predicting. Well, first of all, you're not predicting heads or tails. You're predicting the probability of seeing heads, for example. And so, if you have seen more heads, then yes, you are more likely to predict a higher probability. You will predict a higher probability of seeing heads.
But will the baseline change? So the baseline prediction will change because you're adjusting things based on this?
The baseline prediction can change, or it can. So basically, the question is, once is this when I'm training my algorithm, am I also feeding back the outcomes to the baseline? And yes, I am. But it doesn't have to. This is the baseline just makes any prediction, and it can also change over time. It can update its internal state, or it can be fixed. This doesn't affect the results. So this is this is the kind of result we can get in a classification setting. Now, these results extend beyond classification as well. We had a paper in NIPS where we showed a general reduction between multi-class and structured prediction to this setting. And we also had some work on how to apply this problem in the context of regression. So in regression, we are predicting confidence intervals. And we want a 90% confidence interval to contain the true outcome 90% of the time. This is our notion of capturing uncertainty. And if we, here's an example. Here we are predicting, we're predicting the blood pressure for an elderly woman over time under different conditions. So sleep, stress, etc. And the blue dots indicate the actual observed predictions. Now, we can train a Bayesian neural network to make these predictions from some set of features. And because this is a Bayesian method, it will give us a posterior estimate of uncertainty, which we can plot here. We can ask it for the 90% posterior confidence interval, which will take this shape. And we see that most predictions actually fall outside the 90% confidence interval predicted by the standard Bayesian neural network. Now, if we apply our method, we actually can extend these intervals, and here, as it should, only one data point falls outside the prediction interval. So this basically concludes the first part of the talk, and I would like to summarize these results by basically saying that we were able to, so in many applications where we're interpreting genomes, it is important to obtain useful estimates of uncertainty, especially if these are medical predictions. But most algorithms do not guarantee these assumptions, and there's a family of algorithms called online learning algorithms, which are which are robust to changes in the distribution, but they may not provide useful uncertainty estimates. And we have proposed a framework that can take any online learning algorithm and transform it into one that makes useful predictions of uncertainty. So more generally, in this talk, I have looked at two problems that arise when doing scientific when we're basically doing scientific analysis in the context of genomics. And in the first part of the talk, I gave an example of how we can use data-driven approaches to make experiments significantly more affordable and more accurate by augmenting them with data-driven techniques. So these are two ways in which we can assist researchers in doing biomedical research. So more generally, my work touches on both of these themes, which are related to both machine learning and to genomics. So on the machine learning front, I have done other work that touches upon areas of probabilistic modeling, which is similar to what you've seen before. And this involves the work you've seen before, as well as questions in probabilistic inference, which often rely on techniques in deep learning. And often in this kind of biomedical data, we are lacking labels. We don't have enough label data. And so this motivates research into weak supervision. And also, more generally, I'm interested in various optimization properties of these algorithms. So these are questions about how we can train and produce the right model for these tasks. So at the same time, I'm also interested in problems in genomics. I'm interested in taking these techniques and applying them into real genomic problems. And you've seen my work on phasing, but I have also worked on genome assembly and metagenomics, which is assembling many genomes simultaneously. And finally, one of my most recent interests has been in curating the biomedical literature and combining genetic data with other sources of data, including, in this case, text data from biomedical papers. So more generally, perhaps the most central theme of my work is it lies in AI and machine learning. And in this talk, I have discussed how we can use AI and machine learning techniques to solve certain problems related to science, specifically in genomics. And these are tasks related to experimentation and to the interpretation of genomes. In the future, I would like to build upon these results in order to also address even more practical problems related to human health. I would like to look at how we can use genetic data in order to solve medically relevant problems, which include the following. Now, I want to argue that now is an especially important time to be working on these problems. On one hand, AI and machine learning techniques have matured to the point that we can have get really good performance on large and complex data sets. At the same time, we are about to obtain these data sets from various efforts that are underway, which include the Precision Medicine Initiative and other similar initiatives in other countries as well. So these results will generate massive amounts of data, and they will include both genetic data and other types of data, which can include both functional genomics data, as well as text data, clinical records, papers, and other sources of data, including medical records and sensors. Integrating this data will be a very important research problem that I also want to address. And this will require developing new techniques that can incorporate various sources of data. And of course, this will all be driven by research on core machine learning problems, which include probabilistic modeling, weak supervision, machine learning safety, interpretable models, etc. Research in these techniques will be motivated by the applied problem in science and health, and will also these new techniques will provide more efficient solutions to problems in science and health. And more generally, taking an even higher level, I want to make the argument that even though AI has made great progress in applications in vision and language and speech, the greatest impact of AI on society is yet to come. I want to argue that the most impactful applications of AI and machine learning are yet to come, and they will happen in fields like health, science, and sustainability. These are important problems for which we will soon have large data sets. And the greatest impact will come by developing new techniques that will solve these problems and make progress on problems that matter. And so this concludes my talk, and I'm happy to take any questions. Thank you.
Yeah, great, thanks so much. Just a quick question on your work with the extended haplotyping or the phasing. Were you in the process of kind of reconstructing ancestral genomes and haplotypes? Could you identify any trends in recombination points and breakpoints? And what can you kind of learn from doing that in the future?
So the question is, was I able to identify recombination points while I was working on the genome phasing project? So I wouldn't say that I was able to identify new recombination points. So there's an understanding of where recombination occurs in the genome. So there are so-called hotspots, which occur more often. And this is something that we took into account when we were designing our models. So we can obtain maps of where these hotspots occur, and we can use them to define the probabilities that govern this mixture of latent genomes that I have described. So we have used these results in our work, but I wouldn't say that we have discovered new hotspots because it doesn't really kind of depend on this particular work that we were doing.
So another question about phasing. So you might imagine that you could want to study something like rare genetic diseases with this phased genome information. And I wonder, you talked about the assumption in your model that the genomes are mixtures of these known already phased genomes. So I wonder, in the case of something like a rare disease, is that still a reasonable assumption? And if it's not, how would you know? Kind of, what would your model tell you that you could kind of determine that it wasn't a good assumption anymore?
So the question is, if you have some kind of rare genetic marker that only you have, can you recover its phase? That is the question, yes?
Yeah, or if you want to study something like a rare genetic disease, is this still an appropriate model for that sort of?
Yes, because you're. So first, this data comes in blocks. So first of all, this rare genetic marker will be part of a longer stretch of sequence, which is going to be similar to some individual in your training set. And so even though it differs at this one position, it knows that it's still the same sequence with a small error. So this will be taken into account into the model. And so you will still be able to reconstruct phase in this position because you know that all the rest of the sequence is similar, even though you have this one rare change. So you will still be able to apply this technology even if you have rare mutations or rare genetic markers.
So in cancer, when there are excessive rearrangements of the chromosome, would the phasing technology still work?
Well, no, it wouldn't work in this case, because if you know that your genome has been, well, I mean, so yes and no. I guess it depends on the extent of the rearrangements, but I mean, I would be hesitant to apply it because you know that your genome has been explicitly kind of modified, shuffled to look unlike anything that's in your training set, and so this is not something that you would want to use for cancer specifically. Yes.
So I have a question about your proper cost functions.
Yes.
Proper losses. A proper loss cannot be local, otherwise it's just logarithm, right?
It cannot be local, otherwise it's a...
If your proper loss cannot be local because that is the KL divergence.
Okay, and how do you define local here?
Local means that your cost depends only on the prediction that you make and not on the outcomes that you don't predict.
Well, it would depend on both the prediction and the outcome.
It has to depend on the whole distribution, not on the outcome, because otherwise it's not useful in decision. It's only useful in inference.
So you're saying the losses depend on the whole distribution of the out.
So it doesn't depend only on the Q of what you predict.
Yeah.
It depends on the whole distribution of the Qs.
Yes. So the definition is formulated that it's formulated with an expectation over, but not over the Q, over the Y. It's also the definition involves the distribution of the Y. Yeah.
So then you have to make an assumption about what the functional form of the loss. So what kind of losses do you use?
I have to make an assumption on.
What kind of loss functions do you use?
Well, in this experiment, it was the L2 function and the logarithmic loss function.
Is that so? You talked about the utility of doing a certain operation, right? Because the doctor gives you a probability. So that is an asymmetric loss.
I'm sorry. Can you repeat again? I misheard.
I'll take it.
I have just one question. So there are a lot of statistical methods for phasing. Can you be a little more clear about what aspect of your method made it more, cost efficient and then computationally efficient?
So there are statistical methods for phasing, as we said. Yes. So but these different methods work for different kinds of technologies. So there are the vast majority of statistical methods for phasing work for microarray data. So in our case, we are using it on a different kind of data that was obtained from this specific technology that we were using. And so we needed to incorporate into the model this prior knowledge about the way that the data is the structure of the data as it comes from this underlying technology. And so this required adding extra variables and extra constraints into the model. But the model is inspired by a result from maybe 15 years ago by Lee and Stevens, which devised a general technique of defining these latent genomes as mixtures. Yeah.
Any more questions? Okay, thank you.